Hey everyone, this is Miss Ivy, your instructor, and we're going to cover Chapter 4 today. Chapter 4 discusses eukaryotic cells and the microorganisms in that category. So let's look at an outline first. We're going to be covering parts 1 through 5. We'll have an overview of the eukaryotes. We'll talk about the structures that are found within eukaryotic cells. And then we'll talk about the three categories of eukaryotic cells that cause diseases in humans. So our overview, eukaryotic cells appeared about 2 to 3 billion years ago. So if we recall taxonomy, we talked about domains. We looked at the taxonomic pyramid, where we started with domain and then kingdom, phylum, class, order, family, genus, and species. So the domains, there's 3 of them, 3 domains. And catch up here. One of them is bacteria. One of them is archaea. And the last is eukarya. And our eukarya domain is where we find our eukaryotic cells. So these are things like animals, plants, fungi, and protists. So again, eukaryotic cells are found in domain eukarya, and that includes the categories of animals, fungi, protists, and plants. These other 2 domains, these are going to be made up of organisms that are prokaryotic, which means that they are prokaryotes and they do not have a nucleus. More on that later. Whereas these, on the other hand, do have a nucleus. Alright, so these 2 domains existed previously before eukarya organisms evolved, and for that reason, eukaryotic organisms tend to be more complex than prokaryotes. They have a lot of additional organelles that we did not see when we did Chapter 3 over our prokaryotic bacteria. So we're going to focus on, when we discuss our various eukaryotes, protists, fungi, and helminths, because those are the ones that primarily cause disease in eukaryotic organisms. So let's look at some structures. This is the basic structures that are found in a eukaryotic cell. Looking at this chart, what it's showing you are, it's outlining what kind of structures can be found externally of the cell boundary itself, and then the internal stuff within the cell. So at the cell boundary, what this is called is the cytoplasmic membrane, just like we talked previously about it in our last chapter. So all cells have this cell membrane. All cells have that cell membrane or cytoplasmic membrane. You can use those words interchangeably. Some eukaryotic cells also have a cell wall, but not all of them. For example, plants have a cell wall, fungi have a cell wall, but animal cells don't, even though those are all eukaryotic cells. So if we move outside of the plasma membrane to the external stuff, these are the appendages like flagella and cilia, and we'll go into those in more details in a little bit, and also the glycocalyx. Internally, there's lots of organelles here. We didn't see very many organelles in Chapter 3 because bacteria, again, are relatively much more simple than these eukaryotic cells. But you'll see the nucleus and its subcomponents, ribosomes. There's various items listed here, organelles that make up the cytoskeleton, and various organelles listed here. We're going to go through those individually, but briefly. Here is a generic picture of a eukaryotic cell. So we can see various organelles that's listed all around the image and pointing to those structures within, compared to our little tiny bacteria cell here, who doesn't really have all those fancy organelles like our eukaryotic cell does. Alright, so let's talk about those individually. So the appendages, these are the things that are outside of the cell boundary. These are typically used for moving. So cilia and flagella are found in some of our eukaryotic cells. So we talked about flagella, I'm pretty sure in chapter 3, because bacteria can have those too. But our eukaryotic flagella is larger and thicker than our bacterial flagella. It's more complex, but it does still serve that motility function. It helps the cell move. Cilia, on the other hand, are hair-like extensions. They're only found in one group of protozoa and certain animal cells, so not as common. But we can look at the difference here in these two micrographs. So we see this ciliate here. It has all that green stringy stuff that you can see around it. That is the cilia. And as we've seen before, these longer, thicker ones are the flagella. Now your textbook goes into a lot more detail on flagella, so make sure that you're taking note of what we're discussing in lecture in this PowerPoint, because I will not test you on all that detailed information that's in the book. So make sure you're watching these videos. Now let's move back to the boundary structures, the cell wall and membrane. Firstly, the cell wall. These are found in protozoa and not protozoa. They are found in fungi, but not protozoa and helminths. Those do not have cell walls in their cells, but fungi do. The fungal cell wall is made up of chitin or cellulose, two examples of starches, and this is analogous to bacteria's peptidoglycan. So peptidoglycan was the carbohydrate that was found in the cell wall of bacteria. But these cell walls, regardless of what they're made up of, they're very rigid. They provide structural support and shape to the cells to prevent them from losing their shape, to prevent them from being pliable like animal cells are. The cell membrane. The cell membrane. So again, all cells have a cell membrane. All cells have this. If you imagine a cell like a water balloon, that rubber that makes up the balloon, that would be equivalent to the cell membrane of a cell. But it's not made up of rubber. So in the cell membrane, it is going to be a bilayer of phospholipids. A bilayer of phospholipids. So bi means two and layer meaning layer. It's two layers of phospholipids. And these phospholipids have a structure that kind of, without showing all the detailed exact atoms and everything, the pictures in the book will look like this, like almost like reminds me sort of like a bobby pin. But it has a head here that is considered polar, is what we call it in biology, and these tails that are non-polar. But essentially, when they're in water, they're going to aggregate in a special way to make this molecule happy, to keep things simple. And the way that they will aggregate is by forming this bilayer of them. So we see one layer up top, second layer on the bottom, and the tails, these guys here, the tails, are going to face each other, and they'll be sandwiched together by the two layers of the heads that are going to be that would be facing the inside or outside of the cell. So here would be the outside of the cell, here would be the inside of the cell, and then we can see this skin of the cell, if you will, is made up of these phospholipids, and there is a bilayer of them, two layers where the tails are facing one another and they're sandwiched by the heads. And so this right here, by the way, is just separating it, showing it to you on the inside, but of course this would be all condensed here in reality. So additionally, there are some other things that we should be aware of that the cell membrane will have. Proteins. Cells will have hundreds, if not thousands, of proteins that are embedded into them. In this picture, they're represented by these purple guys throughout. And also cholesterol molecules, that are these yellow things. These cholesterol molecules allow for the cell's membrane to be flexible. It allows for flexibility. Unlike the cell wall that we were previously talking about, that is very rigid. All right, our next structure that we should be familiar with is the nucleus, and this is what defines a eukaryote. All eukaryotic organisms have a nucleus, while prokaryotic organisms like bacteria do not. This is typically going to be the most prominent organelle of eukaryotic cells, and it contains DNA in it. So DNA is encapsulated inside of this nucleus, compared to bacteria, which did not have a nucleus. Remember, the DNA and the nucleus is kind of in this nucleoid region. It's kind of free-flowing in the cytoplasm, whereas here we see our cell, and inside of it a very large organelle that is hollow that contains the DNA within it. This organelle is also considered membranous. You'll see several of the organelles are, but basically it just means that it's composed of two parallel membranes. There's lipid bilayers again, that is similar to the plasma membrane. We also have another organelle called the nucleolus. It is inside of the nucleus, and it's a site where ribosomes are made. And then last definition here, chromatin, that's our DNA, and again, it is housed within this nucleus. Next, our endoplasmic reticulum and Golgi. So again, our textbook may go into a lot more details on these organelles, but for our purposes, we just need to be really brief. So the rough ER, the smooth ER, and the Golgi apparatus are all essentially a series of membranous tunnels, and it's utilized to transport various things such as proteins and lipids as they're being produced. So very, very short there. We don't need to know a whole lot about that. Next, mitochondria. You might have heard before, the mitochondria is the powerhouse of the cell. And that is true because it is the site of production of ATP, which stands for adenosine triphosphate. That is what ATP stands for. And ATP is essentially like our bodies, or our cells, is our cells' currency of chemical reactions. So in order to do many chemical reactions, we have to have energy, and ATP is what we use to spend that energy. We can't just take glucose and use it to drive cellular reactions. We had to convert that glucose into ATP, and that's the job of the mitochondria. So it extracts chemical energies contained in nutrient molecules and then turns it into ATP. Chloroplasts. These are found in algae and plant cells because they are the ones who are capable of photosynthesis. So these chloroplasts harvest sunlight energy and convert it to chemical energy through photosynthesis. That chemical energy is in the form of glucose. Next, ribosomes, really important. They're protein synthesizers. They make proteins. They can be found in the cytoplasm, or they can be attached to the RER, which stands for the rough ER, the rough endoplasmic reticulum. They can appear inside mitochondria as well as chloroplasts, but essentially the big idea which you really need to know here is that they are the site of protein synthesis. That's very important. We're going to talk about those more later. So multiple ribosomes can also be found arranged in chains. We call these polyribosomes or polysomes. But their structure is like this. They have a large and a small subunit that's sandwiched together, and they are described as by their what they call Svedberg units. So in eukaryotic ribosomes, we have an 80S unit total, and then the large subunit is 60S, and the bottom one, The small one is 40S. Next, the cytoskeleton. The cytoskeleton has a few different functions. It helps to anchor organelles. It aids in moving RNA in the cell as well as vesicles, and it permits shape changes. So you can almost think about it like literally the cell's skeleton, if you will, because just like our skeleton helps us maintain our shape, so does the cytoskeleton for the cell. Alright, so let's move to 4.3. We're going to talk about fungi now. When I talk about fungi, we're not just talking about mushrooms. I know that's probably instinctively what we think about, like the ones we eat, but we got to think microscopic. And microscopic fungi include yeasts and molds, and these cause the mycoses. So by definition, a mycoses is a disease that is caused by a fungus. So what do these look like? These look like this picture here, for example. This is a picture of some yeasts. You can see there's some budding forms throughout. They're growing and dividing, essentially. Their yeasts are unicellular. They're going to be round to oval in shape, and they reproduce by asexual budding. This picture here specifically is of Candida albicans. We'll talk about that one again soon. And this is a picture that comes from your book. So they're great about showing you these illustrations, and this is a scanning electron microscope image of one, but I like to show y'all what they look like under our light microscope, which is what this is, because it's the one that's mostly found in clinical sites. What about molds? So molds are made up of hyphae. And hyphae are these thread-like cells that are found within the mold. So if we look at this right here, this is showing you some hyphae. You can see it's very filamentous, long and stringy cells. And these hyphae lack walls and cell membranes between the cells when they are non-septate. On the other hand, septate hyphae will have a septum or a deviation in between cells, and that's of interest because it is an important feature of diagnosis in a mycoses if we're for identification purposes. So again, molds, if we look at them really, really up close, it's made up of these filamentous cells called hyphae, which can be septate or non-septate, and that is an important feature to determine what mold you have. So these hyphae, collectively, they form a meshwork that we call mycelium. And so you're seeing that right here on this orange, as well as several examples seen here on this agar plate. This agar plate is a dish that has this jello-like substance on it that has nutrients that help this microbe to grow. And so you will see multiple colonies here of molds, and this collectively, each one would be considered a mycelium. Some fungi are dimorphic, and di means two, morph meaning morphology, and morphology meaning their structure, so they can have two structures. And it's dependent upon temperature. Temperature is seen here. So some of these fungi that are dimorphic are capable of growing as a mold at 25 degrees Celsius, whereas they'll grow as yeasts at higher temperatures such as 37 degrees Celsius. And just take note, that's at the temperature of the body. So this really helps these fungi to survive in very diverse environments where there can be drastic temperature changes. So there are nearly 300 species of fungi that can cause human disease, and they are acquired in one of three ways. We can have community-acquired mycoses. This is where they are contracted from the environment. Number two, nosocomial. That's an important word. Nosocomial means it's an infection that was caused by entering a hospital. It's associated with a hospital. You went to the hospital and you came out with a bug that you didn't walk in with. And finally, opportunistic infections are another possible way of contracting these mycoses. Opportunistic infections cause opportunistic infections occur when a individual that normally would not contract this infection gets the infection because they have a weakened immune system. So normally, again, this opportunistic pathogen would not cause disease in a healthy individual. But whenever we have an individual who's immunocompromised, then they are more susceptible to getting sick by these bugs, or in this case fungi, that doesn't normally make people sick. But it is very troubling. This is very concerning. There's actually more deaths due to opportunistic infections than breast cancer. Some of the people that are really susceptible to opportunistic infections include cancer patients, transplant patients, and HIV patients, all of which would be considered immunocompromised. All right. Let's look at a few medically relevant fungi. First one is, again, Candida albicans. And again, we saw that it was a yeast before, but this is the most common cause of yeast infections. They can cause, they can cause an infection in the oral cavity. We call this thrush, so that's what you're seeing in this image here. Vaginal yeast infections, as seen here, all this white buildup would be yeast. And the cutaneous infection of the skin, cutaneous meaning skin. So more commonly, yeast infections on the skin are going to be in places that tend to withhold moisture, such as you see here behind the knees. Armpits would be another example of where it would be more common to see yeast infections. Any place where the moisture is going to remain in the body for longer periods of time due to not being exposed to the air. Next, penicillium. This provides our antibiotic penicillin. So interesting, it's actually a fungus that makes that antibiotic. And that fungi is called Penicillium species. That's the genus, or subspecies, excuse me. But they produce penicillin. This was the first antibiotic used to treat bacterial infections, and now we can, of course, purchase it in a pill form. It can be made synthetically as well, but it inhibits cell wall synthesis. And so since bacteria have cell walls and they get exposed to this penicillin, they will not be able to make any more cell walls. So they won't be able to reproduce if we expose them to penicillin. However, do keep in mind some bacteria are resistant, and that's becoming a growing issue in the medical community. Next, Histoplasma capsulatum. So this is associated with bird and bat poop. 60 to 90% of people who live in the areas surrounding the Ohio and Mississippi rivers have been exposed. And this is one of those that's really dangerous for the immunocompromised population. So what we're looking at here in this picture is the prevalence of the infection of Histoplasma capsulatum by area in the United States. And here we look at this picture, which is showing you what it looks like underneath the microscope that you would use in a clinical setting. And to me, they kind of look like suns. So that's how I remembered that back in the day. Next, we have Aspergillus flavus. This is a contaminant of nuts and stored grains. And it can be potentially concerning because it produces a toxin. And that toxin is called aflatoxin, and it is the most potent known natural carcinogen. And carcinogens are cancer-causing agents, and therefore it's associated with cancers such as liver cancer in developing nations. So here we see what it looks like underneath the microscope, and I included this little guy here, the wacky waving inflatable arm flailing tube man, because what it reminds me of under the scope. All right, Mucor species. These can potentially cause necrotizing infections. Necrotizing means essentially eating away of the flesh. It kills cells. Although most species are intolerant to temperatures found within our bodies as mammals, it's not impossible for them to cause these necrotizing infections. And so here we see one man that's famous for it, Mark Tatum. This is before the infection, and this is after. They call him the man without a face, and it's because he contracted a mucor species infection, and it's called the condition was called rhinocerebral mucormycosis. I know that's a tongueful or a mouthful, but if we break it down, rhino means nose in Latin, cerebral referring to the brain, mucor, mycoses. It's a disease of a fungus and specifically of mucor species. All right, let's talk about a little bit of other stuff besides the medical stuff. There is impacts of fungi in regards to the agricultural. The agricultural impact of fungi tends to reduce crop production because these fruits and vegetables and general agriculture in general can contract fungal infections. And so unfortunately that does reduce crop production. We can also see diseases in domestic animals be caused by these funguses that are contaminating the crops that the livestock would use for food. Fungi rot also fresh produce during shipping and storage. So this is an interesting number here. 40% of our yearly fruit crop is consumed by fungi. And naturally, then lots of research and development of resistant strains of these of this produce is underway. And so here what we're seeing is 2 fungal infections. This is squash if you can't tell. But yeah, so nice fuzzy fungi growing all over it. All right, but we talked a lot about bad things here, but fungi do play a very essential role in our nutrient cycling of the earth. They decompose organic matter that is dying or decaying and return those minerals to the soil or the water. They also assist plants. They have a mutualistic relationship with plants in the root systems to help each other absorb more water and nutrients. Fungi have also been engineered to produce desired products like antibiotics, alcohol, organic acids, and vitamins. And some fungi are, of course, eaten to flavor foods. Now, how do fungi get nutrition? They are heterotrophic saprobes. That means heterotrophic means they acquire nutrients from a wide variety of organic substances. We're also heterotrophic. We consume food. These, however, are sapropic, which means they will get these nutrients from dead organisms or the remnants of them by basically absorbing them. Some fungi are parasitic, which means they're parasites. They'll grow on or in a host and then cause harm. These are the ones that would concern us in a clinical setting. So here we see ringworm and athlete's foot. By the way, ringworm is not caused by a worm. It is actually caused by a fungi. It's just called ringworm because it tends to take on oftentimes a ring shape. And then here we have athlete's foot. Next are reproductive strategies for fungi. That can get very, very complex. The textbook goes into some good details on it, but we will not go into this because it is very, very complex and we could have an entire lecture or even an entire class on it. But do know that it is species specific. So it just depends on the species, and they typically will produce spores at some stage in their life cycle. But don't confuse these with bacterial spores. It's not the same thing. These spores, you can think of them sort of like pollen. It's similar to that, where the spores will be distributed through the wind, for example, through water, through pollinators, even through just individuals, them sticking on individuals and falling somewhere else. But once they find a favorable substrate, they will then begin to grow new fungus. So here what we're seeing, I don't know if y'all ever saw these as kids, but they'd be like these small golf ball sized, they're mushrooms, fungi, and if you step on them, they would poof. So that's a picture of that. You can see all the spores being released whenever she stomps on the fungi here. You can also see the spores here. They come out, they are produced in this mushroom from underneath the cap. So if you ever look at a mushroom and look on the underside, you will try to scrape it. You'll see all these little this stuff, and that stuff, if you look at it under the microscope, is the spores. All right, moving to the protists now. So these protists come from the Greek word for first animals, even though they're not animals. They are single-celled typically. They have our special organelles, so they can do special functions like other eukaryotic cells. They have organelles for moving, feeding, protection. Some of them even have a relatively primitive nervous system to help coordinate their movement so it's not random. Some can move through fluids by means of cilia, those hairs, flagella, the tail, or pseudopods. So that one's new. Pseudopods would be these cytoplasmic extensions. Pseudo means false and pod means foot. So they're like false feet. Here we have Plankton from SpongeBob, and he is a protist. Alright, so these protozoa typically live in aquatic environments. Also soil is possible, and they're free living usually. They are heterotrophic, meaning they consume food. They're scavengers for dead plant and animal debris, or they can even graze on bacteria and algae. Most of them are harmless. However, pathogenic species do exist and may live on the fluids of their host, such as plasma and digestive juices, and cause disease. So let's look at the life cycle next of our protozoa. And again, with this, our textbook goes in a lot of details, but we're going to be very brief. So essentially, there is a trophozoite phase and a cyst phase. And the trophozoite phase is the active phase. It's the active phase. Whereas the cyst phase is the dormant resting phase. So as we've seen with other types of organisms, when conditions are not favorable, it will develop into a cyst phase. It will go dormant. And this dormant organism, this dormant protist, will be resistant to unfavorable conditions such as it being really hot or dry or certain chemicals. And it can also be more prone to being able to be dispersed by air currents, which would not be able to be possible in the trophozoite phase. So that's an important factor in the spread of disease caused by these protists, is their ability to have these two different phases of activeness and dormancy that is easily spread throughout the air currents. Some protozoan groups exist only in the trophozoite phase, but many of them have both cycles. Let's look at a couple examples. Here we have Entamoeba histolytica. This is transmitted orally by contamination of food or water, and it causes amoebiasis, which is an intestinal syndrome. It causes dysentery. This is where we will wind up having severe diarrhea with the presence of blood and mucus in the feces due to this infective organism. It's diagnosed by fecal culture. Isn't that lovely? So that's what the clinical laboratory scientists do in hospital labs, is they'll be sent these cultures of, in this case, feces, to be observed underneath the microscope, and they will be looking in the lab to identify any of these trophozoites or cysts. In this case, not sure why that happened. Bear with me. Oh boy. Alright, sorry about that. Finally got it pulled back up. So, as I was saying, it is diagnosed by a fecal culture because these cysts and trophozoites will pass in the feces and can be identified with a microscope. So what we're seeing here is the cyst phase and the trophozoite phase under a microscope using a light microscope. And this is relatively pretty common, especially in our third world countries. Okay, next, this one's interesting to me. Naegleria fowleri. This is also known as the brain-eating amoeba. So it is transmitted via contaminated water that enters the nose. It's associated with stagnant lakes and ponds, unchlorinated pools, and even neti pots that are unclean. So make sure you're cleaning your neti pots and using distilled water in them and not tap water. But this organism causes something called PAM, and that stands for primary amoebic meningoencephalitis, meaning that there's inflammation of the brain and the meninges, those are the protective coverings of the spinal cord, and it can cause death, within 3 to 7 days, like very quick death here. And how it's diagnosed is going to be through detection of the organism in the cerebrospinal fluid or via biopsy. And 75% of diagnoses are made after death of the patient, unfortunately, because it just takes the patient so quickly. So what we're seeing here is a ring lesion on a brain scan, and it will then progress to hemorrhaging. So here we're seeing bleeding on the brain and ultimately death. Next, Giardia lamblia. This is transmitted through ingestion of contaminated food and water, or even feces. Yuck. But also it can be transmitted via fomites. Fomites are just inanimate surfaces like your bench top, etc. But they cause giardiasis, and this is going to result in GI symptoms. And again, similar to our first one we talked about, the Entamoeba histolytica, it is diagnosed via stool sample, but the cyst and the troph look different. So this is probably one of my favorite ones because I just think the trophozoite looks funny and kind of neat. It's like a kite with a smiley face, sort of, or a balloon with a smiley face. So that's the trophozoite phase and the cyst phase. Another example of a major pathogenic protozoa would be Trichomonas vaginalis, and this is a STD, so it's caused by sexual transmission, and it causes, I see some mistakes here. I think I accidentally must have copied and pasted something because this is not correct. It does not cause giardiasis. That was the last slide. My apologies. It causes reproductive issues. 70% of infected people do not have any signs or symptoms, however, which is concerning. It makes it more prone to being transmitted. When it does have symptoms, it's typically going to cause itching, burning, and redness in the genital area, which will also result in discomfort with urination and a change in the discharge produced by the vagina. Is diagnosed by a discharge specimen that will be then viewed on a microscope, and what we will see is something that looks like this. There's a single flagella at one end and multiple at the other, and here we're seeing the organism in a light microscope. I'll fix that issue in the slide. And hemoflagellates, hemo meaning blood and flagellate meaning they have a flagella. So they their transmission is through arthropod bite, things like as seen below, tsetse flies or the reduvid bug. The reduvid bug transfers Chagas disease. These are also called the kissing bug or assassin bugs, but it can cause a chronic and asymptomatic infection until eventually it will cause complications. And one of the one of the, 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 indicators of Chagas disease is what we call the Romana sign, which is where we'll have unilateral periorbital swelling, meaning swelling in one eye, around one eye. So we can see that in this picture here. You can also see chargomas, like that will pop up like this, these darker reddened areas. And another example of a trypanosoma disease is African sleeping sickness, and it is transmitted through the bite of the tsetse fly. And this is dangerous. It's fatal without treatment. And we can diagnose these type of infections with a blood smear, since they are hemoflagellates living in the blood. So here we see a hemoflagellate. These other little guys that are around, the most prominent cells, those are red blood cells. The little tiny purple things here, here, and here, those are platelets, and this is our hemoflagellate. Next, we have Plasmodium species. They are also transmitted through arthropods. This one specifically is through mosquitoes, and it causes malaria. Initially, malaria looks like the flu, but it progresses and can cause kidney failure, seizures, mental confusion, coma, and eventually death. Because these are found in the blood, we will also diagnose it by a blood smear, and what we're seeing is bunch of red blood cells, but this one, and this one, these are red blood cells that actually have been infected with a Plasmodium species organism in it. And so that's just different phases of their life cycles. We're not going to go into the difference between ringform and trophozoite and the schizonts, but in general, this is kind of something you would see underneath the microscope. How can we prevent it? Things like this. We see this individual has a mosquito net that is surrounding their bed to prevent being bitten by mosquitoes that can cause this disease. Something of interest as well, I guess being a biologist, is malaria is a major cause of morbidity, which means illness, and mortality, which means death, that threatens about 3.4 billion people worldwide. In countries where malaria is endemic, the disease represents a major public health challenge. And so I also wanted to point out something of interest that I teach my biology students is if we look at the demographics here of malaria prevalence compared to the prevalence of the sickle cell gene, you'll see they're very tightly interconnected, and that's pretty interesting because sickle cell alleles are more prominent in those areas because having sickle cell trait, meaning having at least one sickle cell gene, makes red blood cells resistant to the malarial infection. And so that gives a hand to evolution right there, and we can tie that into Bio 1. Pretty awesome. Another example, Toxoplasma gondii. This is ingested, and it is a contaminant of food and water. It causes toxoplasmosis. The symptoms of the infection can vary. It can be completely asymptomatic. It can be severe. Typically, it's associated with cat poop and more severe in immunocompromised patients. So this is why pregnant women are not supposed to change the cat litter because it can also cause congenital defects of the child. And it's diagnosed by an antibody test. And we can see that a rapid test right here shown in these pictures. Cryptosporidium. This is ingested, again, by contaminated food or water. It causes cryptosporidiosis. It's a diarrheal disease. Symptoms can last a few weeks. However, complications can occur in immunocompromised patients, and we would use a stool sample with an acid-fast stain in order to view it under the microscope. But there are immunofluorescent assays that are also available. Okay, moving to our very last section here, the helminths. We're going to be really short and brief with them. The textbook goes into a lot more detailed information, like with life cycles and such, but we're not. So these helminths, they include tapeworms, flukes, pinworms, and roundworms. Our adult specimens are usually large enough to be seen with the naked eye. However, they are still talked about in microbiology because of the way they're typically diagnosed is through the eggs that are passed, usually anyway, through the stool. And those can be microscopic and used to be seen under the microscope to get a diagnosis. So not all flatworms and roundworms are parasites. However, many do live free in the soil and water and are an important part of nutrient cycling. But there are some disease-causing helminths that do need to be discussed. They will spend part of their life cycle in the gastrointestinal tract of an animal, and that could include humans. So our flatworms, these are also known as the platyhelminths. And if you need help remembering why platyhelminths are flatworms, platy, think platypus, and they have a flat bill, and helminths mean worm. So they're very thin. They are oftentimes going to have a segmented body plan, similar to your standard earthworm. However, that earthworm is technically a roundworm because it's thick, but flatworms would be segmented and very thin. They can be divided into two categories, the cestodes and trematodes. And cestodes are tapeworms. They are very long, ribbon-like organisms that are segmented. The trematodes, or the flukes, on the other hand, have more ovoid bodies. Roundworms, on the other hand, are not going to be very thin. They are oftentimes going to be elongated and cylindrical. They oftentimes have an unsegmented body, but not always. And they're also considered nematodes. So what we're seeing here is a few examples here we see the flatworms. So here we see this very, very long one would be our tapeworm, and you can see a real picture of it here. And then we see our ovoid shaped flatworms. And then here we see our roundworms. So this is the real pictures of what they would actually look like. You can see that they are going to be, they're not flat, they're round, hence the name. Now let's discuss their life cycles very, very quickly. The general cycle of life for a worm is they will have fertilized eggs and larvae that's released into the environment, and they will make their way to a host. So whatever host that larva develops in is known as the intermediate or secondary host, and adulthood and mating occur in a final host, a definitive host. So the transport host is an intermediate that can experience that experiences no parasitic development. But the exact process, just like we saw with molds or in fungi, is going to really be dependent upon species. So sources for human infection are contaminated food, soil, and water, or other infected animals. There's about 50 species of helminths that cause disease in humans. They're distributed throughout all areas of the world. We see higher incidence of them in tropical areas. And the yearly estimate of cases is in the billions. And they're definitely not confined to developing countries like many infectious diseases are. And it's estimated that 50 million helminth infections are in North America alone, and that's a conservative estimate. All right, and finally, this table shows some examples of helminths and how they're transmitted. You don't need to memorize this for the exam. It's just showing you, we're just making you aware. All right, you guys. So I will post this video soon.